हेलो स्टूडेंट्स। प्रीवियस टॉपिक में हमने माइटोसिस के बारे में डिटेल में पढ़ा था, सेल डिवीजन के बारे में। माइटोसिस टेक प्लेस इन सोमैटिक सेल्स. You have studied that a parent cell which was diploid divided into two daughter cells which was also diploid, and they were having same genetic features, genetic combinations. The mitosis cell division helps in growth, repair, replacement of the worn out damaged cells. It takes place in growing part in plants. Now we have to study today the next type of cell division which takes place in a special type of cells, that is sex cells or germinal cells. It takes place in the gonads, that is the sex organs. In plants, the male sex organ is androecium, where pollen grain was formed. In female part, it was gynoecium, where ovule is formed. We have to study in human being. It takes place in gonads. The male gonad is testis. The female gonad is ovary, where the gametes are formed. In sexual reproduction, the male and female gamete have to fuse together during fertilization to form zygote. So the gametes must have half the number of chromosome to that of the parent cell, such that after fertilization they again restore the original or diploid set of chromosome, such that in each generation our features will be maintained. We maintain our race of the organism. So today, students, we have to study the meiosis, or commonly we say meiosis, but the correct pronunciation is meiosis. Now, students, meiosis takes place in germinal cells. Meiosis is reduction division. We have studied, that is the number of chromosome in daughter cell is half of the parent cell. Now, meiosis, M-E-I-O-S-I-S, so sometimes we also say meiosis. Don't worry for the pronunciation, because in the examinations you must write correct spelling. So meiosis completes into two parts. Just students, for simplicity, I am going through the flow chart. After that, we will discuss it in slightly detail. Although in ICSE syllabus, remember, in ICSE syllabus, the basic concept of meiosis has to be studied only, in which we will study the main process taking place, that is called crossing over. But for the simplicity, for the proper knowledge, or to make our concept clear, we will study this cell division slightly in detail, such that there will be no confusion in your mind that how does it take place. So students, we will go in slightly detail. Meiosis completes into two parts. Meiosis 1, or also termed as first meiotic division, and here meiosis 2, or second meiotic division. This is the reduction phase. That is, in this part the number of chromosome is reduced to half of the parent cell. Now, whenever the cell division has to take place, then the first phase will be karyokinesis. Now there will be prophase 1, metaphase 1, anaphase 1, anaphase 1, telophase 1, telophase 1, and then the second stage arises, that is cytokinesis. Cytokinesis. Now, students, we see cytokinesis 1. First cytokinesis. The parental cell of meiosis 1 was diploid in nature, but at the end here two daughter cells are formed which are haploid in nature. So, the daughter cell of meiosis 1 will be haploid in nature. Now, this one will go to the meiosis 2. That is, the cells or the products of meiosis 1 will undergo meiosis 2. Again, here there will be karyokinesis, in which there will be prophase 2, metaphase 2, anaphase 2, telophase 2, and then finally there will be cytokinesis 2, and as a result we will have here, again it was taking place in haploid cell, and as a result we will have 4 daughter cells, 2 from each of the daughter cell from meiosis 1. So, the 4 haploid daughter cells are formed. Hence, at the end of meiosis, from one parent cell which was initially diploid, the division takes place in two parts, and finally at the end we will have 4 daughter cells which are haploid in nature. Now, for simplicity, we can again draw it with the help of diagram for simplicity, and again we will study it in slightly in detail. What happens in the first meiosis division, because this is the most important part. The meiosis 2 is similar to mitosis, because whatever may be the condition of cell, parent cell of meiosis 2, like mitosis, the splitting of a chromosome will take place and the daughter cells will be formed at the end of that division, which will have, each daughter cell will have single stranded or single chromatids. Now, suppose again we take here 2n, that is parental cell. When meiosis 1 take place or meiosis 1 take place, or first meiotic division, then we will have diploid daughter cells. Again the two divide by the second part, meiosis 2, similar to mitosis, and as a result we will have this type of daughter cells. So this is the reduction phase. Now, so this is the basic plan of meiosis cell division. Meiosis, as we know that meiosis takes place in sexually reproducing organism, because here the fusion of male and female gamete has to take place, the sperm and ovum has to take place such that zygote is formed. If the reduction of chromosome would not have taken place, then after fertilization the 46 plus 46, that is the diploid cells fertilizing together, the number of chromosome will be doubled. That will take place in next generation again, and hence the number of chromosomes in each generation will not remain constant, but it is a meiosis, due to which the number of chromosome in each generation remains the fix. Now, students, let us start with meiosis, cell division. As we know that it has to take place in two parts, completes into two parts. So the first one is meiosis 1, or also we also say first meiotic division. Meiosis 1 or first meiotic division. Again, there will be karyokinesis. Karyokinesis. Students, it is similar to the previous topics, that is the division of nucleus, or we say that nuclear changes taking place, resulting in division of nucleus. So nuclear changes taking place as a result of division of nucleus during cell division is called karyokinesis. Karyokinesis will complete into 4 stages. Prophase 1. This is the most important phase of meiotic cell division or meiosis 1. All the relevant changes which we have to study according to our syllabus belongs to this phase. So we will study slightly in detail. Prophase 1, that is the longest phase. This is the longest phase. Students, in higher class you will study further in more detail, studying sub stages of meiosis also, meiosis of prophase 1 also, but here we need not require to learn all these sub stages. Like you will study in higher class the sub stages liptotin, zygotin, pachytin, diplotin and diakinesis. These are the 5 sub stages that will be taking place in this, but we need not require to learn all these words according to our current syllabus, but the changes occurring during these phases will be summarized in few points mainly, which are required to be studied, which are required to be defined. So let us start prophase 1. In prophase 1, the first change that takes place, the centrosome divides into 2 centrioles which migrate towards opposite poles. Second, the chromatin fibers shorten, coil and condenses to change into chromosome. This is a similar change taking place. The third, which is the most important change taking place during this period. Now, students, the pairing of homologous chromosome takes place. The pairing of homologous chromosomes take place. This phenomenon is called synapsis. So this term is required to be defined, synapsis. What is synapsis? the pairing of homologous chromosome during first meiotic division or prophase 1 of meiotic division is known as synapsis. Don't be confused with synapse. Synapse will be the word used in nervous system chapter. Here this is synapsis. Now students, here although we have described this term homologous chromosome in previous part, while studying the chromosomes we also studied this part, so we can go through that topic, or if we will go for to that video we will find its clear definitions. But again, since you will have some problem here, so I am again defining it. Homologous, the pair of chromosomes having same shape, size and genetic location, one coming from either of the parent, one coming from either of the parents is called homologous pair of chromosomes. Now, like you see, suppose when fertilization takes place, then through the male gamete, that is a sperm, and the female gamete ovum, 23 chromosomes come out. 23 chromosome. Suppose there is the chromosome, single chromosome, which has a particular type of gene or allele on it, and again from this side also there is the another type of another chromosome having the similar type of allele or gene. When fertilization takes place, then it will be diploid, that is having 46 chromosomes, and these two will be in a pair, that is a paired set of chromosomes having the similar alleles on their position. Then this pair is known as homologous pair of chromosome. So students, in normal condition the chromosomes remain scattered, but during this part, this phase, they come to lie side by side, that is called pairing of homologous chromosome, also known as synapsis. Now, in one pair, two condensed chromosomes are visible, hence they are called dyad or bivalent. Now, this was the first change. Next, we will describe it with the help of a diagram. Just come to summarize the terms mainly here. Next, longitudinal splitting of chromatids take place, but they remain joined at centromere. So in one homologous pair, now 4 distinct chromatids are visible. This is called tetrad. Tetra means 4, tetrad, or also called tetravalent. Now students, in one homologous pair, or the two chromatids of the same chromosomes are called sister chromatids, whereas the chromatids of two chromosomes in the homologous pair are called non-sister chromatids with respect to each other. So next part, longitudinal splitting of chromatid take place, like you see, first of all, suppose just I am drawing the diagram here. This is condensed one chromosome. Now just I am drawing it hollow to represent it. Suppose this is from father side, this is from mother side. Now this is the homologous pair of chromosome, also known as dyad or bivalent. Now during this stage, this splitting take place, means the chromatids get separated, divided, splitted, and they remain joined at centromeres. Now this is the shaded one, that is from the father side. This is, and this is the hollow one from the mother side, this one. Now so this is called tetrad or tetravalent. Now you see again, just I have discussed earlier that the chromatid of chromosomes are called, suppose these two chromatids of the same chromosomes are called sister chromatids, and the two chromosomes of two chromatids or chromatids of two chromosomes in a homologous pair are called non-sister chromatids. Non-sister chromatids. So this is the main point here. So a splitting takes place. Next, the next point. The non-sister chromatids of homologous pair coil around each other for exchange of genetic material. Now, so non-sister chromatids of homologous pair coil for exchange of genetic material, The process will be known as crossing over. Just we will define it again. Crossing over. The point at which they cross or exchange their genetic material is called chiasmata. So, the point of crossing over is called chiasmata. Now, here you can represent it with the help of this diagram. This is shaded one, and this will be the hollow part. This is the point mentioned here. So this point is known as chiasmata or simply chiasma. Now, at this point, the breaking and reunion of chromatids take place at chiasmata, and segments are exchanged. That is, in this part you see, suppose you will draw like this. So what happens here? At this point, at chiasmata, when it breaks and reunite, but while reuniting, they get exchanged. Means this part will come on this side and this part will be joined on this side. So this will be the segment. Like This is the segment, यानी segments are exchanged. This is the basic concept. So if you see here, this is from father side, this was from mother side. So on this part some maternal features are exchanged, and on this part some paternal features are exchanged. So this is the significance of crossing over at this phase. Now lastly, due to mutual repulsion, the chromosomes move apart with their exchanged segments. So this is the basic point, or the most important thing taking place in first meiotic division. Now again we have marked here crossing over. So what is crossing over? We can define it clearly, separately. Crossing over, the exchange of genetic material between non-sister chromatids of a homologous pair of chromosomes is called crossing over. So this is the definition for crossing over. So finally you can see here, that is in prophase 1 of meiosis. First of all, pairing of homologous chromosomes take place. After that, longitudinal splitting of chromatids in each chromosome take place. So in one homologous pair, 4 chromatids are visible, hence it is called tetrad. Then the non-sister chromatids coil around each other, and at the point of crossing, which is termed as chiasmata, the breaking and reunion of segments take place. The breaking and reunion, that is the exchange of genetic material between the two non-sister chromatids, is known as crossing over. Now this major change has taken place. Finally, now the further more changes take place, that is gradually the nucleolus and nuclear membrane disappear. Estral rays and spindle fibers are formed between two centrioles. This is the completion of prophase 1. Now after prophase 1, there is metaphase 1. Now the next stage is metaphase 1. Again, meta means the middle. So in mitosis we had seen that the chromosomes get aligned at the center, at equatorial plane. In metaphase 1 of meiosis also this takes place, but this time there is one major difference, that is the pair of homologous chromosomes, not the single chromosome. Pair of homologous chromosome get aligned at equatorial plane, that is homologous pairs of chromosomes are arranged at equatorial plane. Now each chromosome get joined by spindle fibers. Now each chromosome of pair, homologous pair is joined by a spindle fiber at centromere to their respective poles. Now here student, you see, if we will study for mitosis, suppose we take 2n equals to 4 in case of mitosis, then in case of mitosis the 4 chromosomes get arranged separately at equatorial plane and each chromosome was joined by a spindle fiber at centromere to the opposite poles. But in this part you see, if we have taken 2n equals to 4 means paired set of chromosome, that is 2 pair or overall 4 chromosome, the cell is diploid still now. Now in this part the pairing of homologous chromosome has taken place. Now the 2 pair of chromosomes get arranged, whatever may be the number of chromosomes, they get arranged at equatorial plane in homologous pair like this. This is the major difference. So this is the metaphase, and for metaphase for meiosis 1. This was for mitosis. The chromosomes were arranged singly, but here the pair of homologous chromosome get arranged. So this is the metaphase 1. It can be easily identified. Next, the third stage is anaphase 1. Again, this is the simple one. The spindle fibers contract and pull the homologous chromosome apart. There will be no splitting taking place, unlike mitosis. So, anaphase 1, due to contraction of a spindle fiber, the chromosomes, one chromosome from each homologous pair get pulled apart. So, due to contraction of a spindle fiber, one chromosome from each pair are pulled apart, and they gradually move towards the opposite poles. Like if this is the anaphase stage, then the chromosomes in mitosis, you have seen that a splitting took place and the chromosome, one chromatid from each chromosome was moving apart, but this time the chromosome as a whole get pulled apart. So this is the anaphase 1. Now you can see the number is reducing. There were initially 4 chromosomes, but now 2 chromosomes are moving on either side. Last stage is telophase 1. Now chromosomes, one chromosome from each pair reach to their poles, uncoil, uncondense and change into chromatin fibers. Finally, nucleolus and nuclear membrane reappear around them. around them. As a result, two haploid daughter nuclei are formed around them, producing two haploid daughter nuclei. So at the end of the anaphase, now you see that is the two haploid daughter nuclei are formed, and furrow develops in the middle. Furrow develops in the middle, like here you will draw. Furrow develops in the middle, and two haploid daughter nuclei are formed. Nucleolus. So these are the two haploid daughter nuclei. Finally, completing the telophase, that is the karyokinesis, there will be cytokinesis. And similar to that of mitosis, we know that in cytokinesis, cytoplasmic constriction grows towards the center gradually and divide it into two daughter cells. So two haploid daughter cells are formed. So this is the first meiotic division or meiosis 1. Now the daughter cells of meiosis 1 or meiosis 1 undergoes again second division, which is similar to mitosis. That is, this time the splitting of chromatid from each chromosome will take place, and the two resulting daughter cell will receive one chromatid from each chromosome. As a result, at the end of it, so suppose here it is meiosis 2 or second meiotic division. So in this part you see that is each daughter cell of meiosis 1 undergoes division similar to mitosis. As a result, for producing two haploid daughter cells. So at the end of meiosis cell division, from one diploid parent cell, four haploid daughter cells are produced. So just you see, students, that now the gametes are formed, which are haploid in nature, and they will have to undergo fertilization process. When it will take place, again the male and female gamete will fuse to again acquire the diploid condition. That will again give rise to new organism. So in each generation, the number of chromosome is maintained. So students, now there are some questions. in which type of cell meiosis take place? Then we will answer germinal cell or sex cells. Next, what is the nature of the daughter cell of meiosis cell division? We will answer it is haploid in nature. Next, what will be the number of chromosomes in daughter cell after meiosis in human being? It will be 23, not 23 pair. When we will say the end of mitosis, we will say 23 pair, that is 46. But here we will say 23 because it is haploid. It is having single set of chromosome. Similarly, again, another question. The pair of chromosomes carrying the genes or alleles for the similar characters, the answer will be homologous pair. Similarly, another question. The phenomenon of exchange of genetic material between non-sister chromatids or between two chromosomes of homologous pair, then it is crossing over. Now, this is a crossing over that is responsible for variation between offsprings of the same parents. It also brings about evolution of new species because in each generation some variation take place. These variations get accumulated and in next generation again some more variation will take place. After a very long period, several generations, now entirely different, that is the characteristics are entirely changed from their ancestors and hence the evolution of new species take place. So if we write the significance of meiosis, then we can have some points like first, reduces the number of chromosomes in gametes such that after fertilization again the original number of chromosome is restored in each generation. That will help in maintaining the race of an organism. It maintains the generation of race of an organism. Second, it brings about variation. And third, it helps in evolution of new species. So, students, now I think it is quite clear to you that how will the meiosis differentiated from mitosis, and although we have also discussed in slightly detail, but the terms like homologous pair, synapses, crossing over was essential to understand clearly with the help of diagrams. So this is all about the second chapter, that is chromosome, DNA, cell divisions, cell cycle and other things. Now students, if you study or watch the video carefully, you will be able to answer all the things, all the questions related to this chapter. Now students, if you have watched and liked our video teaching, then please share, like and subscribe our channel, and to receive notification of our latest video uploaded, please press bell icon.